In this video, we will discuss reproduction in plants, and this is for IB section 9.4 for the 2016 starting exam material. And in this video, we're looking at how do plants reproduce, and really what we're focusing on is sexual reproduction in plants. And we'll get to some of the structures and the ways that plants are able to do this here in a little bit. But first, we need to talk about the ability of plants to flower, because flowers and the petals of flowers are one of the things that plants use in order to attract pollinators, as we'll see, in order to be able to reproduce sexually. And so there are some different mechanisms that help to control how plants flower and when they flower and open up to attract pollinators. These things are primarily based off of the temperature and the amount of light. And these two conditions, these environmental conditions, can both influence or affect the expression of genes within the plants in order to cause or to change different aspects of flowering. And so temperature can transform leaf-producing shoot into a flower-producing shoot, and this is the day length is the primary trigger for this. The amount of light inhibits or activates genes that control flowering. It activates the flowering time gene, and the following proteins that are produced activates multiple flowering genes, which then actually cause the plant to flower. And so let's take a closer look at how that process works. It's broken down into two different categories called long and short day flowering that we'll look at a little bit longer, or a little bit more detail here. Day length provides signals for plants flowering. So it's the length of day that provides signals for when they should flower. And there's the two types, the long day and short day. The long day flower in the summer when nights are short, and the short day plants flower in the fall when the nights are a little bit longer. Experiments have found that it is not necessarily the amount of light, but it's actually the length of darkness that is what is actually triggering the flowering. We're going to see how that works. And it's primarily dependent on something called the phytochrome protein. It's a blue-green pigment, and it absorbs wavelength, the light in different wavelengths, and it changes its form depending on what type of light, what wavelength it's absorbing. And so then the plants change their response to flowering based off of the length of day. And so this gets pretty complicated pretty quickly, but we're going to try to simplify it and make a little bit of sense of it. This phytochrome protein in plants is what is absorbing the lights. And it absorbs light in a particular wavelength, and it can change its structure based off of the wavelength that it is absorbing. So the two different structures can either be PR or PFR. And PFR structure, the PFR structure, is what controls the onset of flowering. So we'll get to the flowering portion here in just a second. The two different lights, or the wavelengths that can be absorbed, are red light at 660 nanometers or far red light at 730 nanometers. We talked about during the photosynthesis unit wavelengths being of different amounts. Different wavelengths cause the different colors, and so 660 and 730 are the two that we're looking at. When the phytochrome absorbs sunlight at 630 nanometers, it switches its form from PR to PFR. So it's a conversion from PR to PFR. When it absorbs far red light, it switches from PFR to PR. The PFR form is the active form of the protein, and that's actually what's going to cause the flowering. But the PR is more stable than the PFR. It has a better stability and won't be broken down. And so actually in darkness, when it's dark, the PFR converts to PR. And that kind of gives us an idea of why it's the darkness that's controlling this process, the amount or the length of darkness, because the PFR form is less stable, and so in darkness it's going to break down and convert back to the PR form of the phytochrome protein. The PFR is actually what's causing flowering. So that kind of hopefully comes together and makes sense. So flowering falls into two different categories. We have short day plants, or SDP, and long day plants. In short day plants, the receptor inhibits the transcription of genes needed for flowering when PFR binds to it. So when this PFR protein binds to the genes, it acts as an inhibitor, and it inhibits the gene that's going to cause the flowering. At the end of long nights, so when the lights are really long, there's very little PFR remains, because we know that that breaks down into the PR, or it's less stable, so it breaks down into the PR form. So inhibition fails and the plant flowers. So when there's really long nights, there's lots of time for that PFR form to break back down into the PR form. And so the inhibitor fails, and so then the plant is able to transcribe those specific genes because the inhibitor is not blocking that gene being transcribed anymore, and so then the plant flowers. The plants that flower only if the period of darkness is longer than a critical length, a certain critical length. If darkness is interrupted for whatever reason during that long period of darkness by a brief red flash or some flash of light, the plant will not flower, and it's not going to be able to flower. Long day plants, on the other hand, they cause an accumulation of PFR because the PR is converted to PFR, which remains at the end of short nights. So during the day, it's accumulating light, absorbing light, and it is a transition from PR to PFR. Well, at the end of very short night periods, hasn't been enough time in order for the PFR to break down into the PR. And so then the PFR in long day plants actually promotes transcription of the genes needed for flowering. So in this case, in long day plants, the PFR, the high amount of PFR, acts as a promoter for transcription of those flowering genes. So a large presence of it then causes the flowers to actually flower. So then it's also possible to manipulate when plants flower, and this is often done by the presence of some light during the middle of the night, as well as ensuring that the humidity and temperature and other conditions are fixed. But the Siam tulip is an example of this, and it can actually be caused or forced to flower by providing a brief flash or a little bit of light during the middle of the night in the off season of when it typically would flower, which is usually in the rainy kind of spring season when there's a longer day. For the rest of this video, we're going to look at the actual reproductive structures and how plants are able to reproduce. And obviously plants aren't able to move physically like animals are, and so they've had to adapt and evolve some different structures and some strategies to help them to be able to reproduce. One of the things or tasks that you'll be asked to do is to be able to identify different reproductive structures within the plants, and they're outlined here really nicely in this image. Flowers that have both male and female parts are called perfect flowers, or they're hermaphrodites, and so there's different parts that make up both male and female on the same plant. The petals, right here, purple in this case, are colorful and are used as a way to attract insects or animals to help the flowers be able to pollinate. So let's take a closer look at the female parts that make up the pistil. And the pistil is composed of the ovary, where the eggs develop and the seeds actually mature. The style, so here's the ovary, the style here, a slender column that the pollen's going to move down, and the stigma is the upper surface of the style that's going to receive the pollen. The male parts are called the stamens, and they consist of the filament, this portion here, there's 4 of them in this image, and the anther, which is right here, and this is what contains the pollen. The pollen is like sperm in an animal, and then that's actually what's going to get spread by the different pollinators. Inside of the anthers are pollen sacs in which pollen grains develop, and then those obviously spread. Different accessory structures form the non-reproductive parts of the plant, such as the petals to attract pollinators, the sepals, which are the outermost green leaf parts, kind of usually towards the bottom or underneath the petals. The process of pollination is transferring pollen from a mature anther to a receptive stigma. And this obviously has to be conducted between plants of the same species and also within the same time frame. And so one way that plants have been able to do this is through mutualistic relationships between pollinators, specifically usually insects or birds. And so the advantage of this is the pollinator, the insect or the bird, gets some sort of food, and then the plant benefits because it's able to spread pollen from one plant to another to reproduce sexually. Some self-pollination can occur. That's pollination on the same plants. From anthers of same flower or flowers of the same plants. Cross-pollination via mutualistic relationships is more common with insects and birds. This can also sometimes be induced by wind, occasionally water. Most oftentimes, though, it's insects or birds. And so again, the insect or the birds are gaining some sort of food, usually nectar, and then the insects and the birds are transferring the pollen. And so this mutualistic relationship evolved with specific pollinators. And so some plants have petals and structural arrangement that are conducive to only specific birds or insects being able to land on them and to pollinate them. And then that helps to ensure that the same species of plants are being pollinated by the same species of pollinators. So in the process of fertilization, it's the fusion of male and female gametes to form a zygote. And how this works is the pollen lands on the stigma and it moves down through the pollen tube here and it eventually gets to the ovule. The sperm from the pollen are going to fertilize an egg cell. So one of those sperm fuses with an egg nucleus forming a diploid zygote, and the other fuses with another nucleus, which triggers the formation of a food store for the embryo. Because as that seed develops, when the seed actually gets to wherever it's going to grow, it needs some sort of food source, some sugar basically, for it to be able to grow until leaves emerge and the plant can start photosynthesizing. Well, how do these seeds get spread away from the original plant? It's possible that they could grow right in the vicinity of the parent plant, but oftentimes they're carried away by a couple of different means. And one of those can be wind. You've probably seen dandelions. You can spread those very easily. Water is another way to disperse those seeds. And animals are also a great way to disperse those seeds. You think about fruit. It's in a way to attract animals to come and eat the berries or the fruit and then go somewhere else, and then after digestion they poop out those seeds in a different location. Little stickies, seeds that like stick to the coat of a dog or fur of a dog or maybe your socks or something like that can get dispersed. So plants have developed lots of different ways to be able to spread the seeds, and wind, water, and animals are most often the means that they do that. Seeds are very compact, they're nutritious, they're relatively lightweight, which makes them easy to be able to move around. To finish up this video, I wanted to show you a couple different parts that show the structures of external and internal of some different seeds, both of a dicot and a monocot. The corn seed is a monocot and the bean seed is a dicot. And so we've got our seed coats. We've got our epicotyl and hypocotyl. The epicotyl is the embryo shoot. The hypocotyl is going to be the embryo stem. The radicle, which forms the root of the plant, and then the cotyledon, which is going to become the first leaves of the seedling. So then a monocot only has one leaf, and then a dicot has two. That wraps up our discussion of plant structure and reproductive strategies.